Thank you for watching this video on how best to study for gene expression 2. I'm not going to go through the whole lecture. I'm just going to give you the high points that are very important for you to know. You need to know that RNAs are protected from destruction, both in the nucleus and in the cytosol, because they are in secondary structure. They have proteins attached to them. They have their ends modified both in tRNAs and mRNAs. For example, the 5' cap, which is unique for mRNAs, and the poly A tail, and then the binding proteins such as poly A binding protein and cap binding protein further help to protect those RNAs from destruction. Base pairing protects the RNAs, as does blocking the ends. This slide, you just need to know that we have to charge each of the tRNAs to make a tRNA with an amino acid attached to it so that we can use it in translation. This slide, you need to know that we actually have a name for this loop of tRNA at the bottom that binds the mRNA. It's called the anticodon loop. You will never have to really know how to read an anticodon loop, but it's the complement of the mRNA sequence. It helps to drive the positioning of the amino acids after we have charged the tRNAs with their amino acids so that they're binding to the proper place of our RNA. That's how we read the RNA sequence and turn it into a protein sequence. We always read the RNA sequence in 3 nucleotides at a time. That's called a codon. You need to be able to read this table and utilize it. You need to know that the first letter of each codon is found in this first column. You then go to which block has the same second letter across the top. So for example, if we're using AUG, we go to this series of rows, this column, so we're in this box, and then we look for the AUG, which is in the last row. AUG is also the start codon for every protein in humans. You do need to know that AUG is for methionine, UAA, UAG, and UGA are stop codons. You should be able to read and utilize this chart without problem, starting with a template strand, starting with a coding strand, or starting with an RNA. You should be able to use this chart to hand translate any RNA sequence. You do need to know that if you have mistakes in your codons, this can cause mutations in the protein. Sometimes those mistakes don't change the amino acids incorporated, so we say those are silent mutations. If they add a different amino acid, we call it a missense mutation. If it creates a new stop codon, we call it a nonsense mutation. And if it changes the reading frame, that is an insertion or a deletion that is not a multiple of 3 nucleotides, then it actually changes the reading frame and changes the sequence from that frame shift through the C-terminus of the protein. Again, you must be able to read and utilize this table. We did a lot of practice during class to help you do that. Remember that we have to prepare our RNA for translation, and we do that through the process of pre-initiation. It begins in the nucleus just like transcription. It requires a series of proteins that all begin with EIF4, that is an I, EIF4, and then the various subunits of EIF4. The ones that are going to be most important for us are EIF4F and EIF4G. You need to know that the poly A binding protein is required to be bound and connected to the EIF4 complex prior to removing the RNA out of the nucleus. It also requires binding the methionine tRNA. That's done in conjunction with EIF2, and it also EIF3 helps us to bind the small subunit of the ribosome to EIF4G complex. This complex then starts near the cap and scans until it finds the first AUG. Then it's ready to be transported out of the nucleus. Again, you have to take your transcript. You need to add the proteins that bind the 5' cap and the poly A tail. Those are held together with EIF4G. Cut binding protein is also known as EIF4F. These three proteins are critical for nuclear export. They recruit another protein called EIF3, which helps to load the small subunit of the ribosome. EIF2 helps to load the first tRNA onto this complex. Note that EIF4G is going to become very critical in regulation because it is the point of regulation for turning on apoptotic genes, and it's very important for many viral infections. So make a big star around EIF4G that you need to understand all the variations that EIF4G is capable of carrying out. These are just the slides for how initiation and translation work. It is important that you know that we add the tRNAs into the A site. They then move to the P site, and then they are ejected from the E site in the ribosome. That's about the level at which you need to know this. You will not likely be asked to know these other initiation factors or any of these drugs that are shown in this slide. Again, all of the symbols that are in red are the eukaryotic or human names. The things in black are the names in bacteria. Make sure that you understand that the first amino acid produced is always the N-terminus, and in humans, that is always methionine. Make sure you understand that the last amino acid added is the C-terminus of the protein, and that after the protein has been produced, we can do a lot of things to modify our protein. Both Dr. Peterson and Dr. Moore have talked to you about all of these different events. They happen in other regions of the cell, and I will not go into them. But you can proteolyze it, you can add disulfide bonds, you can add sugars to it, you can add phosphates to it, you can even add lipids to the proteins once they are translated. And make sure that you understand this region. This is that how to read the nomenclature. If it's a capital E followed by a number like E2F, those are transcription factors. If it's TF, then Roman numeral, then a letter that's a factor in transcription. That's a transcription factor. If it's EIF with a number, that's a initiation factor for translation. If it's EF, then the 2 is after. That is a elongation factor for translation. And if it's TF with a number after, that is a termination for translation. Make sure that you can identify whether I'm saying E2F, EF2, or TF2. Those mean very different things. So make sure you understand everything in this table, and you can identify factors based on the type of numbering that is used. You do need to know that mutations are taken advantage of in the translation event. Many antibiotics, as you saw in those slides, act on the ribosome, particularly on the ribosome of the prokaryote invader as opposed to the eukaryotic one. There are ones that also act on eukaryotes. There are substances like ricin, which actually can also altering the function of the ribosome. The initiation factors can also be mutated. If the initiation factors are mutated, you get a variety of different diseases. These are the ones that you need to know, that the ribosome can be mutated, EIF2B can be mutated, EF3 can be mutated, EF4G can be mutated. All of those cause various forms of disease. You do need to know the diseases associated with each. You also need to know that EF2, that's the elongation factor 2, can be ADP ribosylated, and that also changes the ability to do translation because it locks up the ribosome and prevents it from moving the tRNAs out of the P site into the E site so that you can move along the messenger RNA. Okay, in the last hour, we actually covered many fewer mechanisms. I mostly gave you how we regulate replication, transcription, and translation. Remember that In humans, we use this system where we take the DNA and do this high regulation of making the transcripts until we can make proteins. We can do it by a variety of ways, by episomal structure changes, as well as second messenger changes, splicing changes, all sorts of ways in which we change how much and which forms of each protein we get out. The most important part of this lecture is how do we regulate these processes in all three: replication, transcription, translation. The amount and availability of nucleotide triphosphates like ATP and GTP is incredibly important. The reason they are important is that these molecules are used over and over again at every stage. Just to put the ribosome together requires 2 units of energy. For every step of adding a single amino acid to a protein, you're using 4 equivalents of energy at a minimum. That means this is a very energy costly event. This is the cycling of elongation, and even this step is also requiring 2 additional units of energy just for us to go from one site to the next. We can regulate transcription by putting proteins onto our DNA. That binding of proteins to the DNA in either replication or in transcription changes the spacing of our nucleosomes. It can actually cause our nucleosomes to be more compact or less compact. As it becomes the least compact, that is when it is typically more likely to be used for either replication or transcription. Remember, the origin recognition complex does exactly the same as these transcription factors do in this slide. You should also remember that we regulate by either simple binding of a transcription factor and its repressor protein. This combination causes the protein transcript to turn off. This is very much like E2F with RB protein binding, preventing the gene to be turned on. If RB could be removed or GAL80 is removed by some other methods, remember RB is taken away when it is phosphorylated. That allows the other half of the enhancer to actually now become an enhancer and turn the gene on. Remember, enhancers binding sites can come in 2 flavors, simple and looped, but we can also have regulation of binding of transcription factors to the TATA box, and we can have these enhancers and repressors that regulate everything. This is just to remind you that many enhancers actually have a binding site a very long ways away from the actual transcriptional start site, and when that happens, we get these large loops of DNA. These various moieties include the transcription factors and the enhancers. Remember that all those external signals that Dr. Peterson talked to you about act directly on the end product of something magic happening in the nucleus. The way that happens is something in their cascade actually ends up binding the DNA. For example, cyclic AMP binds a protein called cyclic AMP response element binding protein. That then binds the DNA. After it is phosphorylated, it helps to then load the transcription factors in the RNA polymerase to start transcription. Likewise, internal receptors, steroid-type receptors, will bind their hormones in the cytosol, usually right underneath the inner leaflet of the membrane, and then they will pick up that hormone, and the hormone receptor complex is transported into the nucleus where it binds its region of DNA and turns on the gene. Remember that maturation of the RNAs is highly regulated as well. The capping is not so regulated, poly-A tailing is not so regulated, but the splicing is very tightly regulated, and it leads to alternative protein structures and alternative protein functions. And we need these other factors as well. We're now going to talk about RNA stability RNA stability, I'm going to give you several examples. Interference miRNAs actually are made and they actually bind to the mRNA. They then guide specific nucleases to come in and destroy this RNA. Because they cut it internally, then this becomes extremely susceptible to destruction by the exonucleases throughout the cell. You do not need to know much more than the fact that miRNAs simply mediate the destruction of transcripts out in the cytosol. This is a diagram that walked us through how an intermediary metabolism protein from the TCA cycle named aconitase actually changes its function when it is low in iron. It becomes a stabilizer of some RNAs like transferrin, because transferrin RNA is very important for the import and stabilization of iron into the cell. Whereas ferritin, which is a storage molecule that holds it within the cell, would not be needed at the time when we have low iron. That same protein makes one of the transcripts unstable. In another transcript, it makes it stable, both in response to low iron. However, when high iron is present, it changes the conformation. This iron-centered protein goes back and becomes a protein in TCA cycle. The stem loop system that it binds, also known as an IRIS system, now changes its behavior without the protein attached to it. Now we flip which is the stable transcript and which is the least stable transcript. So now the one that was unstable to begin with, ferritin, becomes stable so that you can take the high iron and store it within the cell for later use. And the transferrin, which scavenges iron out of the bloodstream, is now not able to be as stable. So we are taking up less iron when we already have high amounts of iron in our cell. Remember, this is all about mRNA stability. This whole slide is just showing you a method for mRNA stability. In this particular case, this is the human antigen R binding protein. It has a specific binding site that is an adenine-rich element. That adenine-rich element also binds a protein called K. When K protein is sitting on the RNA is very unstable and the RNA will be destroyed. When HER is activated, it now binds onto that same sequence. K is pushed off and becomes phosphorylated, and now the transcript becomes stable in the differentiated cell. Notice this is the common way in which we increase the stability of transcripts in muscle or any other differentiated cell. This particular example is for a muscle-specific protein called myogenin. You need to know how these three biological agents act to change transcription or translation in our cells. Poliovirus has what we call an IRIS sequence. An IRIS sequence is simply this stem loop sequence. In the case of polio, two things occur. It has this sequence and it has an RNA viral genome. And so as soon as it invades your cell, it wants to start translating its own genome and make new copies of itself. It railroads the host system by proteolyzing EIF4G. It is cut so that it can no longer hold on to EIF4F. Then it becomes an IRIS binding protein. When it binds the IRES or IRIS, then now the ribosomes can load here instead of having to load at the 5' cap, since this transcript does not have a 5' cap. The IRIS binding then allows this to become the preferential RNA for translation in the cytosol throughout infection. It's an interesting note that human genes that also have irises are also proteins that are involved in apoptosis. Many of the apoptosis genes do have irises and do not get 5' capped. By the time we realize that we are not getting our EIF4G recycled into the nucleus, and what is there cannot bring out 5' cap transcripts, then the virus can railroad and cause us to begin to undergo apoptosis that causes the blebbing of the viral particles out into the environment. So the virus takes advantage of the apoptotic normal signaling system. Ricin is a molecule that comes from castor beans. It's highly poisonous because it breaks a single glycosidic bond, removing the adenine from a specific region of the large subunit of the ribosomal RNA. This specific adenine is required for the binding of elongation factors. If it is not there, that adenine is removed, then elongation factors cannot bind. If they cannot bind, they cannot help the ribosome translocate. Therefore, we have an abrupt stop of all translation under the influence of ricin. And lastly, diphtheria toxin interferes with the elongation factor 2 directly. It ADP ribosylates the active site of the elongation factor 2. This prevents the elongation factor to catalyze the GPT required for translocation. Therefore, again, we completely stop translocation and we stop the cell from being able to translate. Thank you so much for listening. Make sure you know all of the things that are mentioned in this short video. Thank you.